A very warm good morning, guys. Welcome to yet another super cool, super awesome, and absolutely amazing session from your favorite and my favorite BioPoint Stream the World of Bio. Once again, welcoming you all back to our yet another live session from BioPoint, the last minute super revision for the sweet NEET 2021 batch NCRT extract biological classification one shot. Are you all ready, guys? Give me thumbs up on the screen if you are ready for this session. Good morning, Calvin. A very warm good morning. Okay, then, so let's move on with this session. And once again, reminding you that on February 5th, we are launching the Learn With Me, Let's Crack It in Telegram group, in our Telegram group of World of Bio. So, once again, welcoming you all to this super cool launch of BioPoint Learn With Me, Let's Crack It on February 5th. Okay, then, moving on. This is the day one portion for the Learn With Me physics, units and dimensions, chemistry, hydrogen, biology, body fluids, and circulation. This is the portion for the day one crash course. Learn with me, let's crack it. Yes, Calvin. And one more launch that we are going to start is the weekly revision marathon, that is a one session in a week comprising of question discussion. One session in a week comprising of question discussion. These two is coming launch and these are the upcoming launch of BioPoint, and the portion for the weekly revision marathon is starting on February 8th. Its portion is the living world, biological classification, plant kingdom, and animal kingdom. This is the portion for the weekly revision marathon, along with BioPoint Stream the World of Bio as a YouTube live. And the other one, Learn with Me, Let's Crack It, is with the Telegram group World of Bio. So, hope you all are perfectly fine with the two upcoming launches of the BioPoint. And once again, reminding you, if any new member is watching this, do not forget to join the Telegram group t.me/neat_biopoint, where we conduct daily session updates, regular discussion, daily polls, and interact with me. You can interact with each other in the Telegram group. Clear with that, guys? So let's move on. Once again, reminding you, do hit the like button, do share it with your friends, do subscribe the YouTube channel BioPoint Stream the World of Bio, and do not forget to remark us by leaving a comment in the comment section. So everyone, let's rule the world of bio and let's get started with the chapter Biological Classification NCRT Extract. Ready, guys? Ready for this session? Okay then. So Aristotle's classification. Aristotle was the first to classify these organisms as plants to trees and shrubs and herbs, and animals to those which have red blood and without red blood. Animals having red blood cells and animals that do not have red blood cells, and he classified plants to herbs, shrubs, and trees. This is what the classification of Aristotle is. Okay, and Linnaeus brought out the two kingdom classification, that is comprising of the kingdom Plantae and Animalia. This is the classification brought out by Linnaeus. And what were the major drawbacks of the classification brought by Linnaeus? Prokaryotes and eukaryotes were placed under plants. Both prokaryotes and eukaryotes were placed under plants. Unicellular and multicellular organisms were put under a same group. In the same group, it comprised these unicellular and multicellular organisms, and there is no differentiation between fungi and plants. No fungal and plant differentiation. This is what we say to be the drawbacks of the two kingdom classification put forth by Linnaeus. Okay then, and after this is the five kingdom classification, three kingdom and four kingdom. Just leave it. We are moving on with the NCRT extract. That is the last minute super revision, and we are moving only with the most important topics of this chapter. Okay, and the five kingdom classification, which is put forward by R.H. Whitaker. R.H. Whitaker put forward the five kingdom classification. Clear with that? Major characteristics of five kingdom classification. So, what were the five kingdoms that were classified by this R.H. Whitaker? R.H. Whitaker classified organisms into five kingdoms that were Monera, Protista, Fungi, Plantae, and Animalia. Monera, Protista, Fungi, Plantae, and Animalia. These were the five kingdoms that classified organisms. And once again, one thing that I want to say to you right now is that most of you were complaining that there is no sessions for the NEET right now, and we are focusing on the 10th standard. But what happened is that when we come up with a live session for the NEET batch, nothing is there. No one is attending the session. After wishing us with the good morning, they have left. That's what happened, and that's why without any people, that's why we are just moving back the sessions of NEET. And again, we have came up with two upcoming launches, but no responses from you all. Clear with that? So if anyone is watching this, kindly text me in the Telegram. Okay, so moving on with the characters, that is the characters that were used for the five kingdom classification: cell type, cell wall, nuclear membrane, body organization, and mode of nutrition. These are the five characters that are used by R.H. Whittaker for this five kingdom classification. Monera, that is cell type, it is prokaryotic. Cell wall is composed of polysaccharide and amino acid. Nuclear membrane is absent in case of moneyrans. Body organization is cellular. Mode of nutrition, they may be autotrophic or heterotrophic. Clear with that? This is all about kingdom Monera. Protista, they are eukaryotic organisms present. Cell wall is present in some prokaryotes. Nuclear membrane is present in case of prokaryotes. Except monerans, all the organisms have nuclear membrane. Except monerans, all others are eukaryotic organisms. And the mode of nutrition, autotrophic and heterotrophic in case of Protista, and the body organization is cellular. Clear with that? Okay, Calvin. Okay. Fungi, the cell type is eukaryotic and the cell wall is chitin and polysaccharide. Chitin and polysaccharide is the cell wall. Cell wall is composed of what? Chitin and polysaccharide. And the nuclear membrane is present. Body organization multicellular and composed of loose tissues, and their mode of nutrition is heterotrophic. Since this is a table, we are moving like this. Okay, we are gonna move in detail with each and every topic of this. Clear with that? Plantae, it is eukaryotic. Cell wall is made up of cellulose. Nuclear membrane is present. Tissue or organ level of organization. Organ level or tissue level of organization is present. Autotrophic organisms. That is, they have the capability to prepare their own food. Animalia, it's eukaryotic, absent, cell wall is absent, nuclear membrane is present, tissue, organ, and organ system level of organization in case of Animalia kingdom, Animalia. Clear with that? And the mode of nutrition is heterotrophic, and the mode of nutrition is heterotrophic. Okay, then moving on with kingdom Monera. Moving on with kingdom Monera. They are unicellular, colonial, or filamentous. They may be unicellular, colonial, or filamentous. They are unicellular, that is composed of a single cell. They may be colonial, that is, they may be found in colonies, and or filamentous, like, right? Yes, guys. So they are the most abundant microorganisms. There may be four types: coccus, spherical coccus, rod-shaped bacilli, comma-shaped vibrium, and spiral-shaped spirillum. Right. So first we are these are the four types of bacteria, and now we are moving on with archaebacteria. Archaebacteria, and once again reminding you one more thing, the cell wall is composed of peptidoglycan, that is the amino acid, plus polysaccharide that we have discussed, the sugar. So this amino acid and polysaccharide together known as what? Peptidoglycan, right? And this peptidoglycan is composed of two components. What are they? NAM and NAG, N-acetyl muramic acid and N-acetyl glucosamine. N-acetyl glucosamine. Clear with that? And cell wall is made up of peptidoglycan for except in case of archaebacteria and mycoplasma. Mycoplasma lacks cell wall. Right, and these are membrane bound cell organelles are absent in case of these monomerans, and presence of 70s ribosomes. 70s ribosomes are present in case of these monomerans. Okay, guys, is there anyone watching this session? Okay, so archaebacteria means those bacteria that live in the extremities of conditions, like what what we say in case of that, methanogens, thermoacidophiles, halophiles, all these are examples of archaebacteria. So they are the oldest. One thing that you have to bookmark is the archaebacteria are the oldest living fossil. Oldest living fossils. And halophiles, they live in these are the archaebacteria that live in salty areas, Thermoacidophiles live in hot springs, and methanogens in marshy areas and the guts of ruminant animals. And these methanogens are the bacteria that produce biogas or the methane. Clear with that, guys? Okay, so their cell wall lack two components known as muramic acid and peptidoglycan. Peptidoglycan and muramic acid are absent in their cell wall. Okay, guys. Eubacteria, that is the next type. They have a rigid cell wall and a flagellum. Photosynthetic, they can be photosynthetic autotrophs. Example, cyanobacteria having chlorophyll A. What type of chlorophyll is there? Chlorophyll A. Cyanobacteria, blue-green algae colonies have gelatinous sheath and some fix nitrogen. They fix nitrogen with the help of specialized heterocysts in nostoc and anabena. Clear with that, guys? Oval-shaped specialized cells that help in fixation of atmospheric nitrogen is known as heterocysts. And eubacteria comprises of the first component is the PPLO. PPLO. Comment me right now, what is this PPLO? Full form of PPLO. Its other name we can say it as mycoplasma. And comment me right now, what is this PPLO? Full form of PPLO. Yeah, comment me right now. Comment me right now, guys. PPLO refers to pneumonia-like organisms. That is what we say to be the PPLO. Okay. Reproduction in bacteria. The reproduction is mainly by fission or spore formation. Under unfavorable conditions, only the bacteria reproduce by spore formation. All other cases, the bacteria reproduce by fission. Clear with that, guys? Okay, so the mycoplasmas are the smallest living cells having no cell wall. They lack cell wall and they are the smallest living cells. Okay, so they are resistant to penicillin, but tetracycline can inhibit their metabolic pathways. So these mycoplasmas are resistant to the antibiotic known as penicillin, whereas which can inhibit their metabolic pathway, the antibiotic known as tetracycline. Tetracycline can inhibit their metabolic pathways. Clear with that? Next is the kingdom protista. That these all are the important points of kingdom monera. And further questions we will be discussing in what? We will be discussing in the weekly revision marathon. We will be doing that. Clear with that, guys? So, kingdom protista. These are the single-celled eukaryotes comprising of the single-celled eukaryotes. Some have flagella or cilia. Reproduction may be asexual or sexual, that is cell fusion to form cell fuses to form the zygote. Clear with that? This is what is the kingdom protista is. They are considered as garbages of the plant kingdom. Kingdom protista is considered as the. After this session, tell us what is all about the Learn With Me plan. Sure, Calvin, sure. So, kingdom protista comprising of, is also known as or is also considered as garbages of plant kingdom, right? Or garbages of the entire five kingdom classification. Okay, because all the waste are dumped onto this protista, all those things that cannot be included in fungi, that cannot be included in plantae, that cannot be included in animalia, all these are just dumped onto this kingdom protista, and therefore it is known as garbages of the plant kingdom. And their movements may be with the help of flagella, pseudopodia or cilia. They move fastest with the help of the locomotory organ called cilia. Clear with that? So, the first classification of this kingdom protista is chrysophytes. Chrysophytes comprising of the diatoms and the desmids, Golgi algae or jasmids. Clear with that? So, let's move on. And discover what is this diatoms. They are known as the jewels of the kingdom protista. Jewels of the plant kingdom or jewels of the kingdom protista is what diatoms. Diatoms is the jewels of the plant kingdom. Okay, so their body is made up of silica. They have a siliceous cell wall, that is their cell wall is made up of, and their body is made up of silica. Clear with that? So they consider their body is like in the shape of a soap box consisting of two halves, an epitheca and a hypotheca. All these things we have did in this session where we have to explain. In the detailed lecture is there. Detailed lecture has been taken for this biological classification, including all these points. Okay, and the cell wall of this diatoms is known as what? Frustules. So what are these frustules? They are the cell wall of this diatoms. Okay, and these diatoms are the chief producers of the ocean. Chief producers of the ocean. They are known as the chief producers of the ocean. Clear? And leucosin is their reserved food material. Leucosin is their reserved food material. Dinoflagellates, they are mostly marine, photosynthetic. Cell wall is made up of stiff cellulosic plates. Most have two flagella. Red dinoflagellate, that is the Ganilox present in the red tides. Okay, Ganilox causes the red tides, releasing a toxin known as saxitoxin. Right? The sea appears red, that is the red types, and it is caused by this Ganilox or the red dinoflagellate. The third one is the euglenoid. The third one is the euglenoid having a rich protein layer that is the pellicle and two flagella. Photosynthetic in sunlight and heterotroph in darkness. Example, euglena. They synthesize their own food in presence of sunlight and they are heterotrophic. That is, they depend on other organisms for their food in darkness. Clear with that? My God. Yeah. Okay, fine. Slime mold is the next category. Next classification is the slime mold. And one more thing that we have to say in case of this dinoflagellates. Dinoflagellates, some dinoflagellates show bioluminescence. Bioluminescence. That is Noctiluca. Example is Noctiluca. Example is Noctiluca. Clear with that, and this Gonilox causes the red tide, that is a marine dinoflagellate, and they releases a toxin known as toxin. Next is the euglenoid that we have discussed about, that they show mixotrophic mode of nutrition, photosynthetic in presence of sunlight and heterotrophs in darkness. The point of attachment of the toe flagella is known as paraflagellar body. Clear. And their reserved food material is known as the starch or paramylon. Paramylon or starch is the reserved food material of euglenoids. Clear with that? And they have a contractile vacuole, a sexual reproduction by means of what? Longitudinal fission, and sexual reproduction is still unknown in case of euglenoids. Next is the slime mold. They are the known as the false fungi. That is the saprophytic protista or false fungi. Under suitable conditions, they form an aggregation or plasmodium. They form an aggregation or plasmodium. Unfavorable conditions, plasmodium differentiates floating bodies bearing spores. Do plasmodium differentiates into floating bodies bearing spores. Protozoa and protista, they are heterotrophs, predators or parasites comprising of amoeboid protozoans, including amoeba and entamoeba, that is move and capture prey by pseudopodia. Sorry guys. Flagellated protozoans, they have flagella. Parasites causes diseases like sleeping sickness, example trypanosoma. Ciliated protozoans move by cilia, example paramecium. Sporozoans have infectious spore-like stage known as plasmodium. So these are the protozoan protists. Just have a glance with this. What example? Yes, Calvin, the food of reserved food material of euglenoids is not there in the NCRT, but just have a look at that. Okay, the reserved food material is starch or paramylon. Clear with that? And this trypanosoma that we have discussed, flagellated protozoan, they causes a NEET question, sleeping sickness. Trypanosoma causes sleeping sickness, etc., etc., etc. Okay, and what else? Plasmodium vivax, malarial parasite. Okay, the vector, malarial vector parasite is the Plasmodium vivax. Okay, then let's move on. Next is the kingdom fungi. Once again, kingdom fungi. Except yeast, fungi are filamentous, saprophytes, some are parasites. Cell wall is made up of chitin and polysaccharides. They have hyphae, that is the thread-like structures of their body, mycelium, that is the network of hyphae. Okay, so let's discuss about the kingdom fungi. They are echlorophyllous, that is, they are saprophytes. They cannot prepare their own food. They depend on other organisms, that is, dead or decaying organisms for food. Clear. They may be unicellular, that is in case of yeast, edible in case of mushrooms, non-edible in case of toadstools. Calvin, are you here? Calvin, are you here? Okay. So hyphae are of two types, that is sinusoidal hyphae and septate hyphae. Continuous tubes with multinucleated cytoplasm is known as sinusoidal hyphae, and having septae or cross walls is, between them is known as septate hyphae. Is that clear, Calvin, till now? Is that clear till now? Till now, is it clear? Since I'm not getting much response in this class, I'm moving bit fast. When I too know that. Even if I'm not talking, I will be getting what bored with this session. That's why I just what else? Moving on with a little bit speed. Okay, so the plant body is haploid and thalloid, and they have thread-like elongated structures called as hyphae, and interwoven mass of hyphae is known as what? Mycelium. And if the partitions are present in case of this hyphae, it is known as septate hyphae. If the partitions are not present, it is known as the aseptate hyphae or phagocytic hyphae. Clear with that? And reproduction, that is by means of vegetative propagation, fragmentation, fission, and budding. Reproduction by means of sexual reproduction, that is spores, conidia, sporangiosperms, and zoospores. Sexual reproduction by oospores, ascospores, and basidiospores. They are produced in fruiting bodies. They are produced in fruiting bodies. Very sorry, they are produced in fruiting bodies. Taking some notes in NCRT, those extra points that you are telling. Okay, Calvin, okay. Perfect. Okay, then. What else to say? The reserved food material of this kingdom fungi. The reserved food material of this kingdom fungi is glycogen. Glycogen is the reserved food material of kingdom fungi. And the cell wall of this fungi is made up of cellulose. Made up of what cellulose? They are made up of fungal cellulose. That is what chitin. Cell wall of fungi is made up of fungal cellulose, and that is referred to as chitin. Okay, and moving on with the Hobbit's classification of this fungi in the next slide. Yeah, before that, sexual reproduction. Sexual cycle has three steps, that is the plasmogamy, karyogamy, and meiosis. Plasmogamy is the fusion of protoplasm between two motile or non-motile gametes. Two motile or non-motile gametes. Karyogamy is the fusion of two nuclei. Fusion of two nuclei. Meiosis in zygote to give haploid spores. Meiosis in zygote to give haploid spores. Okay. In sexual reproduction, two haploid hyphae fuse. In some fungi, two haploid cells fuse, that is giving two n number, that is the diploid cells. In ascomycetes and basidiomycetes, that is the classification of this fungi. A dikaryotic stage or a dikaryon phase, that is comprising of two nuclei occurs. Such a condition is known as dikaryon, that is including two nuclei. That is what we refer to as dikaryon. Later, parental nuclei fuse to form diploid. Clear with that till now? Clear with that till now? Phycomycetes. Next we have 1, 2, 3, 4, and 5 slides more. Okay, so phycomycetes or lava fungi, they occur in aquatic habitats on decaying wood as obligate parasites on plants. They occur in aquatic habitats or decaying wood as obligate parasites on plants and their mycelium. What is this mycelium, guys? What is this mycelium? Comment me right now. Waiting for your replies. We have discussed that maybe septate or sinusitic in case of protista. Now we are gonna say that mycelium is septate and sinusitic in case of phycomycetes. So what is this mycelium? Comment me fast. Comment me. 10, 9. Comment me. I'm having a countdown for you too. What is this mycelium? 8, 7, 6, 5, 4, 3, 2, 1. And your time's up. So, without under mycelium, yes, what is mycelium, Calvin? Yeah, mycelium is the vegetative part of a fungus, like what or fungus-like bacterial colony consisting of a mass of branching or thread-like hyphae. Oh my God! Again, Abhisar is here to make me laugh. Okay, so mycelium is a branch of a fungi. That's all. Okay, so vegetative part of fungi is known as mycelium, and asexual reproduction by motile zoospores or by non-motile aplanospores, and sexual reproduction by two gametes fusing to form zygospores. Gametes are isogamous or anisogamous or oogamous. I thought, but I didn't tell. Okay. Example for this phycomycetes mucus, rhizopus and albigo. Rhizopus or bread mold and albigo or the parasitic fungi on mustard. I am not at all moving with the minute details because each and every point I am clear that I have covered in the detailed lecture of biological classification. Right. That's why I'm not at all moving with the detailed explanations. Okay. Next is the ascomycetes. Basidiomycetes, and what viruses, viroids. Deuteromycetes are there too. So soon we will complete this chapter. So let's move bit faster to. So ascomycetes, they may be unicellular, example yeast or Saccharomyces. Multicellular, that is penicillium, a source of antibiotics. Mycelium is branched and septate. The vegetative part of that fungus is branched and septate. Asexual reproduction by means of conidia produced on conidiophores. My throat is drying. Sexual reproduction by ascospores produced in sac-like asci, and this asci form floating bodies or ascocarbs. So all these are just basic theories. That's why I'm not at all giving explanations for all these things. Okay, guys? Hope it's very clear for you. So, example, Aspergillus, Claviceps, Neurospora, all these are used, and Neurospora, that is used in biochemical and genetic works. Morels and truffles are edible ascomycetes fungi. Yes, Calvin. Yes, Calvin. I have just planned to take some water right now. After what moving bit more with this chapter. Basidiomycetes. So this basidiomycetes include mushrooms, bracket fungi, or puffballs. Their mycelium is branched and septate. Vegetative reproduction by fragmentation is common. Plasmogamy by fusion of two vegetative or somatic cells results in the dikaryotic structure called basidium, and that after karyogamy and meiosis forms four basidiospores. Clear with that? Basidia are arranged in floating bodies known as basidiocarps, and examples of this basidiomycetes is agaricus or mushrooms, ustilago or smut, Paccinia or rust fungus. Clear with that? Clear with that? Clear with that? And these basidiomycetes are known as the club fungi, right? Club fungi. And they are most advanced and extensively found best decomposers of wood. Best decomposers of wood. These are the best decomposers of wood, and their septa has a clamp connection or a dolypore, or their septum is known as dolypore septum. That is, there is a clamp connection in their septa. Clear? Okay. Next is the deuteromycetes or the imperfect fungi. So asexual reproduction is there. Asexual reproduction is by means of conidia. Only asexual or vegetative phase are known. Mycelium is septate and branched. Some are saprophytes or parasites. Majority is decomposers. Example: Alternaria, Coleotrichum, and Trichoderma. Alternaria, Coleotrichum, and Trichoderma. These are the deuteromycetes or fungi imperfecti. Clear with that, guys? Let me have some water. Till then, please wait. Okay, guys. Moving on with the last and final topic: virus, viroids, and viroids, prions, and lichens. So, viruses are non-cellular obligate parasites. So, they are not at all included in this five kingdom classification. D.J. Ivanovsky discovered virus. Louis Pasteur gave the name virus. Bejernik demonstrated the extract, that is, extracted a fluid from the virus, contagium vivum fluidum, from tobacco mosaic virus, TMV, tobacco mosaic virus. Yes, mine too. Favorite topic is this virus. And W.M. Stanley showed that this virus can be crystallized. So this is all about the introduction of this virus, starting with these four scientists: Ivanovsky, Louis Pasteur, Bejernik, and W.M. Stanley. Okay, a virus is a nuclear protein. There's a protein known as caspid, and a genetic material, that is the RNA or DNA. And guys, one thing that you have to know is that in the NEET examination, almost all the outlines are being covered till now. So just somewhat, just look some inward portions or look some portions deeply from the topics, so that it may ease your scores. Okay, so generally plant viruses have a single stranded RNA. So plant virus have ssRNA, single stranded RNA. Okay, and animal viruses have either ssRNA or dsRNA, that is single stranded RNA, or double stranded RNA, dsRNA. Bacteriophages. What are bacteriophages? Comment me right now. Comment me right now, Calvin. What are bacteriophages? What are bacteriophages? Waiting for your reply. Very simple question. What are bacteriophages? Come on, come on. Bacteriophages are the viruses that attack bacteria. Okay, bacteriophages are the viruses that attack bacteria. Just understand the topics. Yes, exactly, the diagram is there in NCRT. Exactly. So what is bacteriophage? They are the viruses that attack bacteria. Very clear with these topics. Okay, and this bacteriophage usually have double-stranded DNA, dsDNA. Okay, dsDNA, and their caspid is made up of small subunits known as caspomeres. Their caspid is made up of small subunits known as caspomeres. Okay, what is viroid? Viroid is an infectious agent with small RNA and no protein coat. They are discovered by Teodina. Viroids are discovered by Teodina, and it causes viroid causes the potato spindle tuber disease. Potato spindle tuber disease is caused by these viroids. Clear with that? The last slide of the day. The last slide of the day. Prions. Prions, abnormally folded proteins is known as what? Prions. They causes the BSE or the bovine spongiform encephalopathy, or which is also known as the mad cow disease. Mad cow disease in cattle, or the CR Jacob disease. CR Jacob disease in humans. So bovine spongiform encephalopathy or mad cow disease in cattle, and CR Jacob disease or CJD in humans. Lichens is the mutual association between the bacteria, algae and the fungi. Algal component is known as the phycobiont and fungal component is known as the mycobiont. Algal component is autotrophic and fungal component is heterotrophic, and these lichens are the pollution indicators. Lichens are the pollution indicators, and that's all for the chapter. Do not forget to hit the like button, do share it with your friends, and do not forget to subscribe to the YouTube channel BioPointStrip, the world of bio. And do not forget to remark us by leaving a comment in the comment section. Before winding up this session, once again, reminding you that we are launching the super cool Learn With Me, Let's Crack It session on February 5th in the Telegram group. The strategies and how we are going to contact this Learn With Me, Let's Crack It will be informed to you in the upcoming lectures in this live itself. Before February 5th, everything to do with Learn With Me, Let's Crack It will be made clear for you. And everyone, let's stay tuned. And this is the portion for the day one Learn With Me on February 5th. Units and dimensions, hydrogen and body fluids and circulation. The yet another Super Goal launch on February 8th is the weekly revision marathon coming soon from the team of BioPoint. The topics is the covering the first four chapters of biology, NCRT class 11, the living world, biological classification, plant kingdom and animal kingdom. Hope everything is perfectly ready, and do not forget to join the Telegram group t.me/neat_biopoint, and thank you all. Have a nice day. Do not waste your time, precious times. Utilize that in studies. And let's wind up this super cool session. And that's all. Here we have covered the 42nd, almost reaching the 42nd minute of this class after starting this class. And that's all for the day. It's me signing out from BioPoint, seeing the world of bio. Bye bye.